So, welcome to all of you, and today we are going to discuss lecture 3, which is related to basic terms in pharmacy. Okay, so we will discuss the terms which is involved in the pharmaceutical sciences. Okay, so come on the first slide. In this slide, I have mentioned the biosteric A. Okay, so what is the biostere? So you can define this biostere that it is formed by the exchange of atom or group of atoms with another. Okay, and the objectives of a biosteric replacement is to create new compound with similar biological activities, or you can say properties, okay, to the parent compound. The biosteric replacement may be physiochemically or topologically based. Okay, so this paragraph can be defined by taking this example. This is the parent compound. Okay, parent compound means a new species formed from that compound. Okay, so in this, what is the difference between these this fragment moiety and this fragment moiety is the similar. Okay, only the difference is here here five membered cyclic ring is formed. Okay, and in this, if you see here, and this ring is formed by via the cleavage of carbonyl. Okay, so means there is CO bond is formed in this five membered heterocycles compound. Okay, so it is the difference. Okay, and the other qualities of this biostere, these in biostere, the both species means these structures, or you can say these organic compounds, having the similar biological properties. Okay, and this replacement may be physiochemically or topologically based. Topological means structurally based. Okay, so this is the biostere A. Okay, so next we will discuss the biotransformation. Okay, so biotransformation, taking one example, you can see this figure. So in this figure, I have taken the example. If you taken any xenobiotics means if you are taking any drug. Okay, then what happened here? Two phases involved in this. First of all, their transformation into metabolites. Okay, active metabolites. So how active metabolite is formed? There are lots of chemical reactions. Um, Chemical reactions occur in biological environment. Okay, so like oxidation, reduction, and other. Okay, hydrolysis, like that. Okay, so these, by this strategy, these xenobiotics will break down into metabolites. Okay, and these metabolites further link to the any you can say like that albumin or another. Okay, and reaches to the uh, liver. Okay, where there are lots of biological types of enzymes is present. Okay, mainly what happen here, drug is degraded into the liver, in which active compound will be formed in this side. Okay, so since in this there are lots of enzymes involved which is present in the liver. Okay, and there are lots of chemical reactions has been carried out in the biological environment. So this is the biotransformation. Okay, so all the process is carried out in the biological environments in the living organism. So there, this is termed as biotransformation. Okay, next we'll talk about the bioprecursors product. Okay, so what is the bioprecursors product? A bioprecursors product is a product that does not imply the linkage to a carrier group. Okay, but results from molecular modification. Active principle itself, the modification generates a new compound able to transfer metabolic or chemically the resulting compound with the active ones. Okay, so यहाँ पे there is a no need of linkage to another molecules. Only chemical modification takes place in the bioprecursors product. Okay, and through cell modification or enzymatic action in vivo, it is converted to the active moiety. Okay, which is responsible for a particular pharmacological activity. Okay, so next we will talk about the catabolites. So what is the catabolite? It is naturally occurring metabolites. Okay, and next we will talk about the clone. So what is the clone? These are the morphologically similar. Okay, so why it is similar? Due to the same genetic sequence in the DNA. Okay, so you can also define it. A clone is a population of genetically identical cells from the common ancestors. Okay, it is also used a number of recombinant DNA molecules are carrying the same inserted sequence. Okay, due to the same inserted sequence, the morphology is similar. Okay, so you can see this structure, and okay, so it is the clone. Next, we'll talk about the codon. So, what is the codon? It is the actual sequence of three consecutive nucleotide, okay, that occur in the messenger RNA which directs the incorporation of a specific amino acid. You can see this structure. These amino acids involve or incorporate in specific amino acids into the protein, okay, and directs for a particular uh, on termination signal. They stimulates the protein synthesis, okay. So codon, the function of codon is protein synthesis, okay, by attachment through a particular amino acids, okay. So next we will talk about the coenzyme A. So it is actually organic compounds, and it is easily dissociable low molecular weight compounds and non-proteinaceous, okay. Often you can say these are nucleotide, okay, and participate in the either they can accept or donate the electrons or chemical groups, okay. So these are the coenzyme. So next we'll talk about the comparative molecular field analysis, or you can say CoMFA, okay. So what is the CoMFA? It is, you can also card 3D QSAR, okay, methods. In this, different types of statistical correlation techniques has been used for the quantitative relationship between the biological activity of a set of compound with specific alignment, okay, and the three-dimensional electronic and steric properties is also analyzed by this methodology. The other properties such as hydrophobicity and hydrogen bonding also incorporated in this type of analysis, okay. So it is very important for computational chemistry, okay, and other, okay, you can say in the pharmaceutical industries, okay, this type of analysis very important, okay, for designing of new bioactive compounds like that, okay. Next we'll talk about the computational chemistry, okay. So it is a very advanced branch of chemistry, and basically it is a discipline using mathematical methods for calculation of molecular properties or the simulation of molecular behavior, okay. So it is very advanced branch of chemistry, okay. Next we'll talk about the CAD, okay. CAD is also explained by computer assisted drug design, okay. So what is that? In this type of computer aided drug design, uh, involve are computer assisted techniques used to discover, design, and optimize biologically active compound with the propagative use of drug, okay. So in this type of CAD, discovery means their drug development is carried out, you can say, in which these three phenomena include discovery, design, and optimize biologically active compounds with a particular use of drug, okay. So it is very important phenomenon, and it is also studies under the computational chemistry also. So next we'll talk about the de novo design. So what is the design of bioactive compounds by incremental construction of ligand model with a model of receptor enzymatic active site, okay. So according to this model of receptor active enzymatic active site, construction of construction of ligand means they may be a drug or another, okay, is carried out, okay. And these structure of the structure of this is known from the X-ray and NMR data, okay. So it is also a very important techniques, okay. Distomer is a enantiomer is a chiral is a different type. If a chiral compound, what is the chiral compound? If a compound is attached by three four different fragments, then it's called chiral, okay. There are also also other phenomenon which describe the chirality, okay, that is less potent. So distomer is an enantiomer chiral compound but less potent for a particular actions, okay. The definition does not exclude the possibility of other effects or side effects of the distomers, okay. Docking studies. So what is the docking studies? Docking studies are molecular modeling studies aiming to at finding proper fit between ligand and binding site, okay. So docking, in particular docking studies, what is carried out? Interaction is carried out between ligand. Ligand may be either a drug molecule or other biological environment through binding site. Binding site may be either receptor or another, okay. Then double blind study. So in this actually there is a clinical study of potential marketed drug whether neither the investigator nor the object know what subject will be treated, okay. So यहाँ पे क्या है, there is a no information provided for the subjects, okay, in which subjects will be treated. The subject know which subject will be treated, okay, with the active principle, and which one is receive a placebo, okay. Placebo term is also I have defined that it is a type of drug in which all the description is mentioned and if you see the physics of the drug, it is similar to the original, but there is no active compound is added in this. Okay, so it is the placebo. Okay, so next we'll talk about the combinatorial library and synthesis. Okay, so in combinatorial synthesis, in this type of techniques process to prepare large set of organic compound by combining sets of building blocks. Okay, and so you can see this is the simple sets of organic compound, and they combining and give the building blocks. Okay, next we will talk about the combinatorial library. So what is the combinatorial library? A combinatorial library is a set of compounds prepared by combinatorial synthesis. Okay, so library is synthesized by combinatorial synthesis. Okay, so you can see these type of library, there are lots of library can be synthesized by the combinatorial synthesis. Next, we'll talk about the double product. Okay, so double product is a biologically inactive molecule which is transferred in two step, either action of enzyme or chemical. Okay, chemical reactions to the active species. Okay, so it is the any drug. It is the product, and through some modification, and it is converted to the active ones. Okay, or you can say some reaction process or enzymic enzymatic action, it is converted to this one which is active ones. Okay, next we will talk about the congener. So congener is a substance literally con means with generated or synthesized by essentially the same synthetic chemical reactions and the same procedure. Analogs are substance that are analogs in some respect to the prototype agent in chemical structure. Okay, so you can say this can be synthesized by this one and another. This might is also needed for. Okay, so these are the congeners. These are the congeners. Okay. So thank for watching this video, and if you have any queries regarding this, please write down your comment in the comment box. We will solve your problem. Okay, and please, if you like this video, please must subscribe this channel. Okay, okay, because this channel is new, and with your contribution, this channel can be grow. Okay, so thank you for all.